get the thing lined up in the screen.
And I don't think we have any other announcements, except for maybe this might be the last session. We'll see how far you guys get.
Yeah, I'll if I get through what I wanted to get through today with the couple species with the short axilla key, then I think that'll probably be it. And of course, if anyone still wants to, has specimens that they want ID'd, they can always send me images and ask for clarification, on a one by one basis.
I hope somebody brought some for today even. But here you go. Sam's got his scope ready.
All right. That's what I got.
Yeah, that looks like Howard eye to me. So the axilla and the similar species Andrei are a bit shorter. Here they extend pretty much to the end. In this species, they extend pretty much to the end of the posterior margin of the scutellum and even further. And you can see that the bands on the Droege are very clearly interrupted medially. Like they're not even, trying to touch.
Right.
And that's what separates, this species from Epeolus scutellaris, which is, the one that co-occurs with it. And I think both are, late summer, autumn species. When was this specimen collected, Sam?
Let's see. And I think you got one other specimen that you probably have up there of Howard eye from us.
Yes.
And this is 26 August.
Yeah, so that makes sense. Yeah, there's another similar species in Florida, Floridensis, but that tends to be most active in spring. But yeah, nice specimen.
Is it anything about the host? And it seemed like it was a sandy site in both those cases, if I recall the other one correctly.
I think Kalides Howard eye has been identified as the tentative host. I think that was according to Mitchell. Let me
yeah, not known. Howard eye is, I think, like maybe North Carolina, which is weird things have happened, but I haven't seen anything up here.
And I think that's what where the type locality is in either North or South Carolina. No, it's in the type locality is in North Carolina. And let's see what he has here. So Kalides Howard eye was a suspected host according to Mitchell, but that maybe it takes multiple host species.
Yeah.
Or maybe that association was incorrect. And there was another.
Or maybe Howard eye is up here.
That could also be. So yeah, you'll just have to collect more. Let us know.
Yeah. Well, after we finish our compilation, there'll be a long list of targets that we'll try and track down. This says in the side. So Mitchell was a North Carolina person. And even though, like, he grew up in Southern Plains and had, I think, a second home down there, which is right on the border with South Carolina, but he essentially never went to South Carolina. Like, I went to South Carolina on a couple trips to a really neat Sandhills place. I think I got 100 new species for the state. It was just ridiculous.
That's really bizarre.
Depauperate it is.
To be so close and not
yeah
cross, especially when you're revising the bees of all of the eastern United States. You think you might visit at least some of the.
Well, I think a lot of it, I think he during his revision time, he just reeled in everyone's specimens.
Right.
And I think he it was a different era, so I don't know that he was even allowed to go out. Like, state people are usually not allowed to go out of the state in terms of even like all kinds of things. So he had several constraints, but he also had probably a parochialism of North Carolina first or something. I think very close.
That's interesting. I didn't know that. But yeah, so this is one of the I believe this is one of the species that he described. Yeah. Yes. So this is a Mitchell species. So I'll start sharing my screen now.
Or we'll never get out of here.
Okay.
Do you want that glabratus at all or not?
You can pull it up quickly, just in case it might be Zonatus, because
Okay.
then that would take us to another part of the key, because the two are quite similar.
Okay. Yeah.
So yeah, before I share my screen, if you want to pull it up, I can confirm.
That'd be great. And I would have thought on your visit you would have seen this one.
Yeah, that
This
looks
is
like
pretty old.
Glabratus to me.
Okay.
But the easiest way to tell is if you look at the pleura, if they're like sparsely punctate with very smooth interspaces.
Uh-huh. I noticed here's a really nice example of that hook.
Yes, and if you were to draw a line between the tips, you'd see that they extend past well past the mid-length of the scutellum. So yeah, that is a really nice illustration of the hooked axilli.
Yes.
And in the similar species Zonatus, they don't extend so far back posteriorly. They're still hooked, but they're like angled laterally, as opposed to posteriorly. And then
I want to say I collected that on saw palmetto.
It's it might be the most common Epeolus in Florida, in peninsular Florida. So the interspaces are quite well defined and smooth and shiny. They're much closer together in the similar species Epeolus innatus, so this is quite clearly Glabratus. Interestingly, if you're going by DNA barcodes alone, this and Epeolus lactoides would come out as a single. You would conclude that they're a single species, but no one's ever, come to that conclusion based on morphology because they're quite different. Glabratus has very much reduced pubescence on the metasoma, whereas in lactoides there are distinct bands, and T1 and I believe T2 are almost entirely red in Glabratus, as are the axilli and mesoscutellum. So and of course the two species are separated by geography, with Glabratus only found in peninsular Florida and a thin strip of coastal Georgia, and lactoides occurring elsewhere in the eastern United States, including the Florida Panhandle, but not the peninsula for some reason. So it's kind of it's another example of that that phenomenon of Hymenoptera with extensive red coloration in peninsular Florida. But yeah, I agree with your IDs there, Sam.
All right. Whew. All right. Well, I need to get more Epeolus, obviously.
Yeah. You don't have bifasciatus, do you? Or
well, I think I do. I think we
okay. That's a very quick one to ID, so we can just pull that up because it's very distinctive. And I think it's one of the ones that if you go by iNaturalist too, like most of the Epeolus records from the United States are Epeolus bifasciatus. Really
very different.
Yeah, it's in a species group that was at various times thought to constitute a separate genus, Trophocleptria. And they're largely wasp mimics, the members of that group. So this one is appears to be a eumenine mimic of some sort. You might, someone not familiar with or not overly familiar with bees might conclude this to be a a potter wasp of some sort. But they're definitely Epeolus. The length of their axilla is somewhat borderline. It just I scored it as just meeting that threshold of extending to about that cutoff of 2/3 the length of the axilla, although sometimes they get a bit longer. So that they key out in the first half of the first third, I guess, of the key. But the very obvious diagnostic features are it's the only Epeolus in North America with north of Mexico with two bands on the metasoma, only a basal band on T1 and only an apical band on T2. So it kind of gives the illusion of an elongated T2, which is characteristic of various potter wasps. Now, in some specimens from Florida, I think the basal band may be missing, but the very obvious feature that sets this species apart from all other North American species north of Mexico is the presence of two well-defined tubercles on the frontal area, on the frons. So if you look at the face, yeah, and this is a good thing to bring up on the microscope because they're more obvious when you rotate it and move it around than not necessarily so clear in images. Yeah, that's beautiful. So yeah, you can very clearly see those two tubercles, and this species is has been tentatively associated with Calides latitarsus, which is a physalis specialist. So whenever I find a patch of Physalis heterophylla, that's the species that's common in my area, I always look around for those two, and I get Calides latitarsus is pretty common here in the Ottawa area, and I've collected quite a few Epeolus bifasciatus in places where there's a lot of Physalis. So,
we do too. It is
I think
heterophylla. Is that a annual Physalis or a perennial?
I mean, I think the individual plants, like here they die back, but I don't know if they're coming back from rhizomes or from roots, or if they're just completely new plants that have seeded. But I think they're, I know the exposed leaves and flowers and fruits all die back, but then come back from the same spot year after year. And they often send, I think, really long runners. So they'll pop. So even if you've got a small patch in your garden, they'll show up in a completely different area. But I think, heterophylla is the species we have here. And I think, is it longifolia might be the one that's more common where you are.
And that's what we have. And I tried to grow it, but it was an annual one, like I forget the scientific name. It could have been heterophylla because I'd read about it, but it's an annual and was like cut leaf ground cherry, and it didn't seem to have any Calides latitarsus on it. And tomatillo, I've heard conflicting things. So I grew tomatillo right next to the others, and there was nothing on the tomatillo and tons on the longifolia. So it might be a preference. Then nearby, someone else was growing tomatillo and had them on it. So
Lawrence Packer grows tomatillos in his garden, and he's had Calides latitarsus go to them. The other species of Calides that we find that I get sometimes on Physalis is Willistoni. And I think that one might be the host of Epeolus interruptus, but I've gotten Willistoni here. I haven't gotten the Epeolus yet. But then the clammy ground cherry Calides heterophylla. I also get Perdita helictoides. There's
helictoides.
Yeah, there's a Perdita.
I said Nova.
Yeah, but it's a nice example of a floral specialization, and an Epeolus that is kind of, even though Epeolus themselves as adults, they'll go to various different flowers for nectar, and a lot of times you'll find these on asters. They're still confined to the same, at least as larvae, confined to the same diet as their hosts are. Right. And so these, in a sense, are also specialized on Physalis. But I just wanted to bring this up as an example because it's a very commonly observed Epeolus species, and it's quite different, in overall appearance from others. So there are, once you get into Mexico and Central America, there are a lot of species that look like it, and this one can be very hard to distinguish from another species, Epeolus obscuripes, but I have not seen that one in the US yet. So as far as I know, there's only one Epeolus in this Trophocleptria group that's found in the US, and that's bifasciatus.
Yeah. I'll point out that Physalis makes a wonderful garden plant. People think of it as weedy, but you put it in a big planter, And the longifolia that I have here makes just a wonderful mound. It blooms for a really long time, and it's just circled by Latotarsus all the time. It's just real fun.
And heterophila, the fruits are edible, and they're actually quite tasty.
Yeah.
So I don't know
think
about
most of them are edible. Yeah, but
Yeah.
they're small in longifolia.
Yeah, they're big, but they're very sticky or larger, but they're very sticky in heterophila. But yeah, it's an example of an underappreciated pollinator that pollinates something that actually we consume. And I think, like other solanaceae, these plants have to be sonicated. But before I go off on too much of a tangent,
right. Okay.
If you can, I'll take over and start sharing my screen soon. Okay. So is my screen visible now?
Yep.
Okay. So yeah, what I thought I'd will key out today, we can start with, since we're in the East, let's do Epeolus canadensis, because it's fairly widespread. It's it's one of the more commonly collected species with short axilla, although it's not as common as species like bifasciatus and scutularis. But I think it's a good candidate. So let's start with the couplet describing the length of the axillae. Let's see if I can remember which one of these might be canadensis. Yeah, oh, that's pretty good. Okay. So the character is axilla in the dorsal view with the tip extending to less than 2/3 the length of the mesoscutellum. So this is what it looks like in canadensis. You can see that it's, they get to about the halfway point, but definitely not past, not to the 2/3 point or past. So this is what I would consider short axillae. And if you're not, if you're new to this character, it would be good to measure directly using a calibrated micrometer within the eyepiece until you sort of get the hang of it, because an unexperienced person might say, oh, these look kind of large to me, but what I refer to is how far back they extend posteriorly. So in this case, clearly we take, we go to jump ahead to 21, couplet 21. Yeah, this might be sort of hard to see in images of live specimens, especially when they're shown at different angles, but it helps when, can help when people upload different pictures of specimens. Also a good thing to do on iNaturalist. It really helps experts identify things if you post more than just a single image, because sometimes the critical shot, will be missed. But yeah, in this case, you can still kind of see that the axillae are relatively short. So scroll down to 21. Doesn't look like the actual couplets are hyperlinked. Okay, so this one's pretty easy. Does the vertex have two pairs of shiny impunctate protrusions? There's only 2 species in North America that have this. Wait, sorry, I pulled up the wrong image. So this is not the same. These are not the same protrusions found on the fronds of Epeolus bifasciatus. Instead, here I'm referring to 4 protrusions found on the vertex. And there's only 2 species in North America that exhibit this feature. And you'll only find them between Arizona and Texas. So yeah, I'm not sure if I have a very clear shot of the head of Epeolus canadensis. Not here, and it's probably a little hard to see. Whoops. Oh, yeah, you can kind of see there's not, yeah. So in this shot of the head, the image is a bit blurry, but I don't see any well-defined protrusions here. So that eliminates those 2 species.
And usually there's not even really hints either.
No, and sometimes there are some species that have wherein there is kind of a bump here on the side, but they're never, and so they have these, you know, what could be interpreted as lateral protrusions, but they're usually not shiny and impunctate like this, usually not as well pronounced, and they're missing the ones by the ocelli. So there's only sometimes a single pair, and they're usually not so well defined. So that is the other. Yeah, let me see if let me just reread this couplet in thorough. So head with vertexillary with 2 pairs of shiny, usually impunctate protrusions. Okay, I had it was without protrusions, but yeah, there may be species where you have a little bit of a bump to the side that may be interpreted as a protrusion, although I think maybe they come out, or if they would be in this part of the key than in later parts, which is probably why I just said vertex without protrusions. But in any case, Epeolus canadensis doesn't have, you know, any. So, or not even a hint, as Sam mentioned. And what was the other feature? Okay, and the T2 fascia with 2 pairs of anterolateral extensions. So what I mean by that is we talked about there being, you know, a lobe-like extension on each side in each species. Well, these ones actually have 2 of them. So yeah, sometimes it's not so well developed, but instead of a single pair, you'll have 2 pairs. Let me see if they're, I don't know if they're more visible in diadematus. Yeah, in this one that you can see that there's 2 pairs. So canadensis is a species that doesn't have any lobe-like extensions on each side. So here you can see there's just a fascia on T2. And you can look at it at different angles. And you can see there are no lobe-like extensions. So clearly, we're not dealing with these 2 species, Camiseraceae and diadematus. So we'll skip to couplet 23. So this here is where you have some species that have that have those hooked-like axilli, but they're angled laterally. So canadensis is not one of those species, but I'll show you an example of a species that does have axilli that are kind of like hooked laterally. So this is Epeolus origeranus. The axilli don't extend clearly past, you know, to 2/3 the length of the mesoscutellum or further, but you know, there's clearly a hook. It's not like a straight line from the tip of the or from the base of the axilla to the tip. And I'll see if I can find.
I'll point out that the left axilla looks in that picture really straight.
Yeah, yeah, and that's kind of,
and
you have to kind of see the line where follow the line, because yeah, I'm not sure if that's maybe the angle, or sometimes you get specimens in which one side doesn't quite look like the other, and that's when it helps to see more representatives. But yeah, I mean, it's kind of subtle because there's like a little bit of a dip, but in this, yeah, you're right. On the left side, it's not as clearly pronounced as it is on this one.
And
but let's
like
take
you
a look
pointed
at
out before, it's just the angling the specimen a lot can be very clarifying. So you probably do this, but some people, particularly when they start out, they tend to stick it in a piece of clay like we do for photography, and then look at it and then make a decision. But you really want to be using your fingers and using your hands as a platform. We've talked about this in the here, so that you can use the light to angle and show suture lines, and particularly in these dark areas.
Yeah, and let me also see if I could pull up a picture of a specimen where it's a little more obvious. It should be this one, I think. Yeah, Zonatus, I think, is one. Yeah, here's another. Yeah, you can,
okay.
Yeah, so here you can see, yeah, it doesn't extend past 2/3 the length of the mesoscutellum. And it's sort of like I feel like it's angled laterally, like it's forms almost an obtuse angle kind of, but then you have this little hook. And I want to see if I can find, where's my image of close some of these because we don't need them anymore. Let's find canadensis. So this is what the axilla look like in Epeolus canadensis. I think there's also, it's not just that they're hooked, but also the length of the free portion. So in those where it's hooked, it has to be the free portion is about 2/5 of the medial length, if not longer. And that's where you look at the straight red line that I drew through. And here you can see it's clearly, this free portion, it's quite tiny. It's definitely not, if it was 2/5 the length, it'd be somewhere here that it would start to diverge from the side of the scutellum. And you don't see that. So, even if the hook thing throws you off, just the fact that these tips are so short should make it clear that that this part of the key doesn't describe it. Or this part of the couplet doesn't describe it. The other thing is this if you have a female, all these species with the axillae that are hooked laterally, they also have a very distinctly bell-shaped or campanulate pseudopygidial area. So where if you were to draw a line and continue along the edge of it, you would see that it's quite clearly bell-shaped, as opposed to something like this or this, where it's more, forms more of a lunule. So let's see if I have a. Okay, and this is a male, so that feature doesn't work here. But that's another one that you pay attention to if you were keying a female. So yeah, well, the first half of this couplet clearly doesn't describe canadensis, so we'll skip ahead to couplet 29. Okay. So now there are 4 North American species in this group that have a very distinctive small patch of tementum anteriorly on the scutum. And so I'm separating those species from the rest here. And so here I describe it as an anteromedial patch of bright or pale yellow tementum. It's either chevron-shaped, horseshoe-shaped, or V-shaped. Although sometimes it's semicircular. Now, unfortunately, in this specimen, the tementum anteriorly on the scutum is rubbed off, but I'll show you what it's supposed to look like. Yeah, these pictures aren't all that clear. I'll try with a better example. That one. Yeah, so instead of these well-defined lines of pale tementum, parallel lines running along the side of the scutum, you have these like really short little patches that are touching. Yeah, that picture looked, I thought that might be a clearer image, but I'll have to go back to the naturalist.
So they're all kind of restricted to that medial area, and the lateral extent is, like maybe not even half the width of the scutum. Is that?
Yeah, it's very small, and that's an important point because let me go. Yeah, I'll pull up some other species in that group as well. Let's go to compactus.
And also, you're showing a lot of Epeolus, which have stuff going on there in that same area, but maybe always a different shape. They have the 2 lines.
Yeah, I've never seen a Epeolus with this pattern of pubescence. But when you get into South America, there's a whole bunch of Droege that that have a similar sort of small patch. But here you can see it's kind of like a chevron shaped. Does it get any bigger? No, it doesn't look like it. But here you have this sort of like, yeah, chevron shape. Now there are some species. Yeah, I'll read the couplet first in thorough, just so yeah, the mesoscutum with an anteromedial patch of brighter pale tementum, usually chevron, horseshoe, or V-shaped. If it's V-shaped, then it's narrowed anterolaterally. So it gets, what I mean by that is, see, these parts get kind of thinner to a point. Sometimes the patch just like will occupy the whole thing. But if they are like at all separated, then they'd be narrowed to this point. And why that's important is because there is another species, Epeolus minimus, that for some reason in California you get specimens where the paramedian bands are joined posteriorly. Let me see where, if I have, oh, should have just put Epeolus. There it is. So this species is Epeolus minimus. Because, but this is you know, an unusual sort of geographic variant where the bands are somewhat joined posteriorly. I think it's to an extent because they're so very much thickened. So when you do get this and you're not sure if it's, you know, in this species group that Epeolus, to which Epeolus canadensis belongs, or a different one, you can see that these bands are not at all like narrowed anteriorly. They don't come to a point, whereas in the other ones, Epeolus compactus, for example, they are. So I'll pull, yeah, I'll bring up pictures from the key which show it somewhat more clearly. So this is you know, the situation in Epeolus canadensis. You can see that the bands are V-shaped and they kind of come to a point here. And if they are, you know, very much thickened, let's see, you know, then there's no, yeah, so here's an example of a species where it's just like one continuous patch. But if they are thickened, then there's no sort of like, there's no gap here. It's just a big spot. So this is a, you know, an instance where you have to really carefully read the couplet. The only exception, you know, the only species that you might misidentify, you know, as belonging to this group is Epeolus minimus. In all the other ones that key out later, there's a pair of well-defined paramedian bands, and I'll show you what that looks like. So let's see. So I have to find a good example where the pin isn't blocking them. Yeah, so here's an example of, you know, two clearly separated, well-defined bands in Epeolus. This is Brumley. Let me see if there's another example of a species with two well-defined separate bands. So Sam, does that make sense?
Yep.
Is there anything that I should clarify about that couplet? Because it can be a little tricky.
No, I think this is the value of having you here talking it through, because that's a pretty unique feature. Like, it's not like you see and blah, blah, blah. It's like you have to really define it. So it's good.
Yeah. And I'll see if it's visible in this one too. Epeolus splendidus. This is a exclusively southwestern species. Yeah. So here's another example where it's just, in this one it seems to just form like a dense patch, but clearly there aren't two separate bands, and they're not parallel to one another. So based on, so unfortunately with our canadensis specimen, I had to identify it as such based on other features, because the pubescence is clearly rubbed off in this one. But I know that that's the state that it's supposed to be. So based on that,
Right.
I'm jumping to couplet 30. Couplet 30 separates a very distinctive Epeolus from the rest of the members of that group. Epeolus splendidus. Let me see, but let's pull up the, like, there's nothing else that looks anything remotely like this. You know, not just in North America, but throughout the range of this species and into Mexico and Central America. It's a very distinctive Epeolus because it's got these really bright yellow bands of tomentum on the mesoscutum and metasomal turga, and it doesn't have a well-defined dark area on T1, as in Epeolus canadensis. See, there's like a really clear triangular area here that's missing in this species. And so just based on the pubescence, you can clearly tell that this is not like the others. But another interesting feature about this particular Epeolus is that the propodium is like very smooth and essentially, sorry, the latter, the outside of the metapostnotum or the propodial triangle, the propodium is like very smooth and essentially impunctate, whereas in the other three species in this species group, it's you know, you can see there's like distinctive microsculpture that dulls the integument, and there's, you know, it's quite clearly punctate and not glabrous. So clearly our specimen isn't Epeolus splendidus, which
let me just bring up one little thing. So in Molly's key had the same place where you have yellow and you have white bands in Triepeolus banding on the, you know, on the tergites.
Yeah.
But I remember when first going through her key that I'm looking for yellow, and like my mind has yellow as a picture, and I'm like, that's not yellow, that's dirty off-white.
It's very subtle, yeah.
So
unless you have like,
I think, yeah. I
unless
don't know
you
if you're
have like
clarifying that,
Triepeolus.
but it took me a while to really get the difference. And still sometimes I'm like, yeah, I'm not 100%. So
And in
just
this case,
a word that it throws people off
yeah,
who
and
are
that's why
insiders.
having pictures is so important, because the difference here is it's either pale yellow, which is what I say this is, or like bright yellow, or deep yellow, however you want, you know, you might want to call it. But having pictures helps. And yeah, in Triepeolus, yellow is very different. It's like a beige with warm undertones is yellow versus like gray, or like, you know, something that it's like almost like warm versus cool undertones is kind of
right.
like
The useful thing is the contrast between right on a B, like you can see the air quote yellow parts on Triepeolus by comparing it to the other parts of the tomentum on the rest of the body that are clearly bright white. And that helps clarify things because, again, when you're starting out, you don't have a lot of examples and things like that. So you can see that even in the splendens. Even your bright yellow is not necessarily people's definition of bright yellow. In the Triepeolus Epeolus Epeolini world, that's bright yellow, but not necessarily in the artistic world.
And another important thing to remember is that if you're looking at museum specimens, these colors could be very faded. And so I have encountered some, you know, the state of specimens is very important to consider when you're looking at coloration. And because it can mess things up, even looking at integument coloration. I've noticed that really old specimens that have been prolonged to light, they tend to develop sort of a reddish integument coloration. And specimens that were brightly colored when they were caught, if they were exposed to light for any period of time, those colors may fade quite well, where, you know, this difference doesn't become so clear. So it's a good thing to remember when using these keys, that we're assuming that the specimens are in this, you know, in the state that they should be, which is, you know, that they haven't been degraded or in any sort of way, and that they're sort of like collected in the prime of their lives too. Because you can also collect things in the field that they're toward the end of their specimens toward the end of their lives, where much of the pubescence might have come off and become faded just because wear and tear.
Right, they're more like guidelines.
Yeah,
like they see in the movies.
Yeah, I mean, yeah, this is like, if you collected a species that is just declosed and, or a specimen that is just, emerged and is in good condition, this is what it should look like. So, and sometimes, those are the best characters that we have to go on, or the only characters that we might be able to go on. And so, another instance where it helps to have, beg your pardon?
There's no shame in sending it off to Tom or sending a picture or just calling it SPA, or if you can, narrow it down to a group, you can say that too.
Yeah. Yeah. And I think a lot of people, I've noticed in the literature, when people do bee surveys, Epeolus is so rarely collected that for the purpose of statistical analyses, a lot of people just seem to say, I'm not going to bother with this key and just, I will only have a single specimen. I'm just calling it SPA.
Right. Yeah.
But like I said, I'm happy to, identify anything from images. So if you do want to get a name on your specimens, and there's even if it's just one collected in a bee survey, I'm happy to help. So going back to the key, we know that it's not Epeolus splendidus. The specimen clearly looks very different. So this is so we'll start here. So the discal patch, that's, if you remember, this black area on T1 that's kind of surrounded, at least on some, if not all sides, by bands of pale tementum. The discal patch, it's quite clearly triangular or semicircular, and the sides are essentially straight, or they're slightly convex. And that means that the discal patch takes on either a triangular or semicircular shape. I've noticed that in Epeolus canadensis, it tends to be more triangular in males and more clearly semicircular in females, but there is overlap, so that's why that character isn't described in the key. But you can see it quite clearly in the holotype of Epeolus canadensis, what a semicircular discal patch looks like. So it's in many species of Epeolus, there are 4 distinct bands of tementum on the T1. There's an apical band, a basal band, and then longitudinal bands connecting them. In this species, the lateral longitudinal bands are essentially continuous with the basal bands, so you don't really know where one ends and the other begins. And that's a very important thing when keying this species, because in a very similar one, Epeolus compactus, which let me see. Thought I had it up, but maybe I'll have to find it again. No, I'll have to search for it. Yeah, let's bring up because there's different examples, and yeah, this is a good one. So, in the similar species Epeolus compactus, you have an apical band, a basal band, and then you have a distinct kind of angle or band that separates them. It might be very short, but that's what separates that species from this one. Now, every once in a while, you'll find compactus specimens where the bands are so thickened that they kind of almost eliminate that or hide that longitudinal band that separates them. But in those cases, you get more of like a diamond-shaped discal patch, not really a not really a distinctly triangular semicircle one. This one's kind of a yeah. So this is a difficult specimen because you might think that this is compactus, but that's kind of partially because the tementum here is faded anteriorly, and so this should really, go like this. But and the angle too makes it look a little more triangular, but this is actually what I would refer to as a diamond-shaped discal patch. If you're not sure, you can look at the length of the discal patch relative to the width. So yeah, so the medial longitude in canadensis, the medial longitudinal extent is more than a third the lateral extent. So if you were to measure this and this, you can see that this is clearly more than a third of this length. Here, yeah, it gets a bit, it looks more like maybe a quarter. But there are specimens of compactus that can be difficult to tell apart because they have this sort of like, because the bands are thickened laterally and they have kind of almost a diamond-shaped, verging on triangular discal patch. But if you're not sure, if you're collecting in the east, there is only Epeolus canadensis. Epeolus compactus is an exclusively Western species. The ranges of the two do overlap to some extent in the Western United States, but in the Eastern United States and in Eastern Canada, there is only Epeolus canadensis. And the two, even though they're quite similar, they do barcode as distinct, and the morphological differences do hold up. With or are supported by barcodes, and they also seem to parasitize closely related Calides. I think, Epeolus canadensis has been associated with Calides kincadii and Calides eulifii, and I think compactus maybe only with eulifii, but I'd have to double check that. But they do seem to go after similar, if not the same, Calides. So I just want to see if I could pull up some more pictures of compactus just to show you the variation in the shape of the discal patch, because it can be quite different, and that lateral longitudinal band isn't always as well defined. So here's a better example of what I would call like a diamond-shaped discal patch. So it's not like, it's not as clearly triangular as it is in canadensis or semicircular. This is this can be a tough character, and you really have to see series. I think, it really helps to see series of specimens, more so because Epeolus compactus is so variable. Whereas in Epeolus canadensis, the discal patch is always, this clearly either semicircular or triangular. And so, based on that feature alone, that I separate, our specimen here from the others as canadensis, and it's also in a part of the U.S. where I would only find Epeolus canadensis. The range of compactus, I think the westernmost records are in Texas. The easternmost records are in Texas, and then they're found all the way to California and British Columbia. So I'm not sure what else, with less than 10 minutes left, what other Epeolus I might have time to sort of, to illustrate. That was going to be my example for today. Maybe we'll leave it to questions. If anyone has questions. Sam, is there anything that I could better describe? Or
no, I think we've broached all the main, identification morphological features, and have covered some of the trickier ones, and they're all good discussions. So that's great. I don't know if anyone, I think Claire mentioned that people could bring in some pictures. Does anyone have any pictures they want to share? And Tom will lay hands on them and divine their divine the truth.
Yeah, let me stop sharing this on my screen just in case.
Anybody can pop in.
Or chat.
Yeah,
And there's no questions in the
ask
chat
your
right
own
now,
questions.
but good spot too.
Yeah.
Okay. Well,
it does tend not to be a chatty group.
Okay.
I think they're, taking notes, and they're, they're absorbing these kinds of things, but they're mostly looking to us to, talk through things. So
yeah,
very
so
niche classroom.
Yeah, so maybe I'll just a few concluding points then about the key. Like I said, it is based on the assumption that the specimens you have are in prime condition. But there are, useful ways that you can, if you're not sure, read about, the host records, the floral records, look at the known geographic distribution, or just distributions, because, it's a fairly thorough revision. I'm not saying that there aren't gaps that, there are no unfilled gaps, because there definitely are, things that we'll learn, especially for the rarer species like Epeolus andrii, which I mentioned in a previous week. But like for some of the more well-known ones, you can be quite confident that, the ones that you can look at the list of material examined and make a judgment call, and like, okay, he's seen like 50 specimens of that species and they're all from this area. Chances are, what I have isn't that, just based on where the specimens were collected. So yeah, I would use as many tools available to to try to build confidence and IDs. But where a lot of knowledge gaps remain are in host records. There's so much research that can be done for host parasite associations. I think one of the more interesting papers that I put out in recent years was confirming a host parasite association by actually barcoding larvae that were taken from a Calides nest, a nest of Calides consors mesocopus. There were lots of Epeolus americanus in that area, so that, and they were observed entering nests, but, we didn't see specimens directly ovipositing. But, you can take a strongly suspected, association and confirm it by doing things like recovering larvae from nests and barcoding them. So in this case, it worked out and confirmed suspicions and got a small paper out of it. But yeah, those, and if you do happen to come up across an aggregation, those are rare and they're like real gems. So take advantage of the fact. Call me if you find one, or email me, because yeah, there's lots of common species for which we still don't know for sure what the hosts are. We may have strong suspicions, but like Calides latotarsus and Epeolus bifasciatus, that that association is based entirely on collections of the two in co-occurrence with one another. There hasn't been a single case of a larva of Epeolus bifasciatus recovered from a host nest, or a video of a female ovipositing. So that's where a lot of the knowledge gaps remain, or in ecology.
Yeah.
There are still some unresolved taxonomic issues or things that I'd like to better resolve with an Epeolus, but that's more in the perhaps northern Mexico, southwestern US. There's that Epeolus pusillus group that I'd like to be able to barcode more specimens to see if the morphological features I identified hold up, with more molecular data. But the gaps are really in ecology, and this is a great group to to be able to look at host parasite associations, because we know that there's only a single host genus. With other genera like Triepeolus or Nomata, it could be very difficult to identify the host, because you're not even sure if you're looking at necessarily the right genus. But in this case with Epeolus, there haven't been any confirmed uses of other host genera.
So Tom, do you have anything that could be called tips when you're out in the field to find and look for Epeolus? Obviously on flowers, people are just going to collect them, but What about nest searching behavior? Is that a thing at all? Do you see them? Do people find them? Is there
Also
a characteristic
photography.
of that?
Yeah. So
How in the field
sorry,
photography.
what was that, Claire?
And also in that theme, how in the field to do the photography.
Oh, to deal with, yeah. Well, so I find that, I mean, males, actually both sexes you can get on flowers. I kind of look for the hosts first. I look for evidence of Calides, but sometimes you might get a specimen with no Calides around that, like an Epeolus with no Calides around. That sometimes happens, but they tend to be a lot more abundant when there's lots of Calides. So you can get both at flowers. Sometimes the phenologies are a little out of sync. Like for whatever reason, I find that male Epeolus often emerge, a few weeks before the females start to make an appearance. That seems to be common in various groups of bees. Um, and it's the females that you may find sort of hovering closer to the ground looking for nest entrances, in areas where there might not be flowers. But I always scan kind of the ground when I look for things. Areas with somewhat sandy soils tend to be, favored, but you can find them in places with dense vegetation as well. But I like to look for them, in areas where there's lots of sand, road banks, river banks, and if I mean, if you know that the Calides is an oligole, you can look for the plants too. Like I look for physalis patches or solidago. Euthymia is used by all sorts of Calides and seems to be favored. Various asters, and yeah, in terms of I don't spend so much time imaging things in the field, and I find that Epeolus aren't very cooperative. They might, stay at a flower for a split second and take off. But sometimes, for whatever reason, they just, pose for you. But it's much harder to, image them when they're flying around close to the ground. In that case, unless they've stopped somewhere and are crawling around looking for nest entrances, it's hard to get a good image of them. Yeah.
So I think we've had some interns playing around with like getting them in nets, or getting just in general the bees in nets, putting them in a chiller in the field for just a little bit, and then so you can like look at them.
Yeah.
So we'll tell you how that works out.
Yeah. They can cool down and survive quite long. Like Epeolus autumnalis will live in the fridge for many days, for example. So they can be chilled for a good period of time and then released or done, or preserved permanently after. Another thing I do when I look for them, I've I've become a cheater in recent years, and I use iNaturalist a lot when I look for potentially productive sites. And I found, like, as an example, there's this one site in Ontario where like a whole bunch of people have posted pictures of Holcomposides, which isn't all that common here. But like repeated observations, descriptions, and I said, I could use a few in my reference collection. So I,
That's
went
not
over, and sure
cheating.
enough.
Using the resources.
I do that with plants a lot. Yeah.
Yeah. So that's another thing iNaturalist is great for, is if you're looking for physical specimens of things, you can learn of some potentially good sites. I don't think, Epeolus isn't enough of a coveted taxon by collectors that would risk extirpating populations like it would for, butterflies and things like that. But, for now, I feel like it's okay to to do that.
We'll start obscuring the GPS when it's a little more extreme. I'm gonna wrap up the recording at this point, but two people have jumped into the chat, so I'm sorry for the people who are listening to this later, but you will not hear that part of the conversation. Okay. It's done.